welcome Meredith. We appreciate having you here.
Thank you. It's good to be with you all tonight.
Meredith Gale is the Director of Development for Glaucoma Research Foundation, and I think she may have met most of you already. And then
a couple of years ago. Yep.
Yeah, and then joining her is Xin Duan, and Xin is with Catalyst for a Cure, and he's out of the University of California at San Francisco. So welcome to both of you. We really appreciate your time and being here.
Thank you.
Thank you. Before I turn it over to Jen, I wanted to give you all just a brief update, and then I'll do a little deeper introduction for Jen, and he'll give a presentation for about 15 minutes or so, and then we'll have time for questions and answers afterwards. Mike, is it okay if I share my screen?
Yes, absolutely. Jennifer,
I just have
can you pass that to her?
Yes, she should be able to.
I should be able to. Let's see. Okay, great. Thank you all for your partnership over the past years. It has been so wonderful, deepening our relationship. I know several of you all have been ambassadors for us. Can everyone see my screen right now?
Yes.
Okay, great. Well, before I turn it over to Jen, I just wanted to touch on, a thank you for that partnership of over the years. Give you a little information on our Glaucoma 360 Gala coming up in February, where Ron is actually going to be one of our honorees. And then I'll introduce Jen. As most of you all know, our shared mission, there's lots of overlap there. And so it has just been so wonderful, like I mentioned, over the past 3 years to have deepened that relationship. And 2019, when you all made that significant gift of $150,000 over 3 years, it coincided so beautifully with the kickoff to the Catalyst for a Cure Vision Restoration Program, which Jen is going to give us the update on this evening. So it's just been absolutely amazing. In July of this year, our board actually approved the Catalyst for a Cure Vision Restoration Initiative for its second phase, based completely on the merits of their findings, which have just blown away all of their advisors. We expected great things out of this team of 4, and not only did they meet those expectations, but they have far exceeded them in such a short amount of time. And we also wanted to say thank you in partnership. Tosin will be joining our board in January, and I think just continuing to strengthen that bond between our two groups as we fight to find a cure for glaucoma. So here's a little bit of a dive into what to expect at our gala on February 10th. You can see Dr. Fellman will be our Catalyst awardee, and then we'll also be honoring Stephen and Michelle Kirsch with our Visionary Award. They recently made a $1.5 million gift and asked that we use it as a match. So between now and the gala, we'll be raising that matching money to fund the second phase of Catalyst for a Cure. And Dr. David Calkins, who is the chair of our Scientific Advisory Board, will be receiving our President's Award this year. So it should be a really exciting event. And so here we are. Here's our dream team for Catalyst for a Cure 3 Vision Restoration. And with us this evening, you all have heard from Anna Latour and Derek Wellesby before. Young Hu rounds out the 4. He's at Stanford, and this evening you'll hear from Jen. So we're really excited to dive in. Dr. Duan is an assistant professor in the Department of Ophthalmology and Physiology at the Wheel Institute of Neurosciences of the University of California at San Francisco. And the expertise that his lab brings to the team is they investigate retinal ganglion cell subtypes, intrinsics, and the intrinsic factors, and test their roles in optic nerve regeneration and vision recovery. 2 of the big goals that the team has dived into in phase 1 of their research has been to develop a method for transplanting retinal ganglion cells in order to replenish the cells lost to glaucoma. And the second element is developing neuroprotective strategies to prevent loss in the first place after transplantation. So I'm going to turn it over to Jin. I'm sure he will share his screen and he'll be able to do that deeper dive with everyone. And then we'll be able to answer questions from there. So thank you all.
Thank you, Meredith. Let me try to share my screen, make sure this is working. Good evening, everyone. Can you all hear me all right? Like the volume, everything? Good?
Yes.
Okay, great. Okay, thank you so much for this wonderful opportunity to meet you virtually, and I hope to meet more of you in the coming years. And again, very grateful for this generous support jointly from Cure Glaucoma Foundation and also Glaucoma Research Foundation together to support Catalyst for Cure Team 3. So thanks, Meredith kindly introduced all of us. I wouldn't spend the time to tell you the team members, and plus like you probably heard the research presentation from Derek and Anna late in the past cycles. And then what I'm going to do today is have a very brief review of the how the team structure together and then why this is important. And for the majority of the time, I'm going to focus on one story in terms of how a junior team of mine at UCSF and collaborated really with the support from CFC3, the Glaucoma Research Foundation, Catalyst for Cure, to really drive one question home with a lot of support, a lot of collaboration, and that tells you why we need a team together. Okay, so very briefly, this is where we were, and then this is where we are moving to. We were very lucky to finish the move right before the pandemic, but right now it's a matter of getting everything restarted, essentially from Golden Gate Park to Mission Bay campus, and welcome most of you to visit us in the near future. I understand the Harvard was canceled, and hopefully some of the AAO events. So basically, as Meredith just introduced, and for this group of audience and expert, I wouldn't waste the time to tell you what is retina, what is glaucoma. However, I want to highlight that there are three phases of the progress, and then of course, the easiest and then the most intuitive one is to have neuroprotection and regeneration, and eventually cell replacement. So Anna Latorre from UC Davis, and collaboration with all of us, has mainly dedicated to the cell replacement, while my work heavily focused on neuroprotection and regeneration. However, it has a very interesting spin for the following reason, which I'm going to tell you in a few minutes. So particularly in collaboration with the other three groups, the key word has been the retina ganglion cells. And that is our key players here. And also a lot of the effort is really to understand why they're dying, how to protect them from not dying. But eventually, one of the critical questions is about what kind of function we can preserve, and how can we really reverse the death, and most importantly, how to reconnect the retina ganglion cells via the axon regeneration to remake functional synapses. Okay? So as you heard a lot from Anna, particularly Derek, and to protect the existing cells, to protect the existing ganglion cells is very critical. And often we suffer from this, the typical chart where you see from the human in terms of how to protect the existing cells, but also when the cell death happen, how to replace the lost cells. So these two concepts has been back and forth, and as you probably heard a lot of the critical development from Derek and Anna. But when we really look into the protecting existing cells, there is a very interesting spin that comes from very basic research from our laboratory. So that is to say, basically, there are like estimate, right, like a million or less of the cells that's connecting to the brain, but they come in many flavors. And when we think about how our vision is really composed, they're really just like as simple as electrical signals that link the eyes to the brain. However, they really come in different flavors that carry different visual features. A very oversimplified work that really stem out of basic research at my junior laboratories through my past training at Harvard, where we really use genetics and use a mouse as a model organism to understand what are the diversified type of ganglion cells, especially the retina ganglion cell that send the axons to different part of the brain. Essentially, when we use the genetics to track the development, the growth, and the regrowth of the cells, we know roughly the current estimate is there are 30 to 40 different type of ganglion cells out there. Obviously, they're carrying different functions, and however to preserve them, however to restore the function is the key topic today. So when we really overlap all those 30, 40 type of cells together, as you can imagine, this is like a highly overlapped arrays of cells and forming the mosaic on top of each other. And the only way to understand this is really through the genetics and most recently single cell genetics and genomic approach, where we can combine the genetic information with imaging approach, and then we can really lighten up a subset of them, study their individuality, eventually do the synthesis back to the envelope, okay? So with that going, basically we have been using, on showing on the top of the right, is a so-called single cell genetics approach, a single cell genomic approach to dissociate the entire retina, in this case a mouse, and it can also be human donor sample, where they dissociate the entire population and really cluster them. And there are roughly 30 to 40 different clusters that can be molecularly distinguished and also functionally different. And while we understand the diversity of the cells, and this single cell genetics and single cell genomics approach can also uncover what are the molecular components, what are the molecular constituents, such as growth factors, such as transcription factors, such as cell ligands and receptors that are important for the regeneration. And in this case, basically, we can use the intrinsic cues from this discovery and try to put it back. And last but not least, and when I showed you a tiny bit of the cells that grows their axons back to the recipient areas. And in this particular case, and we are particularly focusing on how to use the intrinsic factors to restore the functional synapses. I'm going to have some limited data on this, and given the time of the 15 minutes limit. Close all the notification that's going to disturb the presentation. Okay. So through this collaboration, we have been very fortunate to pick up one of the most effective experimental glaucoma model. That's called silicone oil model, which I'm going to skip a lot of this. And this is, I'm going to leave the opportunity to other team members to tell you. Essentially, from team 1 to team 3 to now team 3, now we have finally have a very reliable and also reversible glaucoma model that allow us to achieve high ocular pressure in experimental animals, including mice, rabbit, and most recently monkeys. Okay, this is all glaucoma CFC3 progress, which we just presented to the board in August. And what is really cool is through the work of CFC2 in the last decade, particularly infrastructure based in Stanford University, where they have this very high resolution imaging apparatus, in this case, experimental animals, that we can lighten up individual cells that we're interested in. In this particular case of cells, it's a Y cell, Y type of ganglion cells, and in the mouse it's called alpha type of ganglion cells that's lightened up with a GFP, and it can also couple with functional units such as calcium indicators, voltage sensors. And we visualize them and really observe their growth and development over a very prolonged time. In this particular case, we superimpose the silicone oil model, which is an experimental glaucoma model, on top of this genetic labeling, where over time, you can really see before and after silicone oil treatment, and before after silicone oil treatment at different resolution in terms of what are the cells that are selectively disappearing. Okay, on the individual cell level, you can see the cells disappearing at certain percentage, but also when you really zoom in, you can observe their structural changes, such as their dendritic arborization in terms of how they are carrying the signals. And all this exciting work in progress that's made possible through the marriage of the CFC3 across 3 or 4 teams, where we can really join forcefully work together that no individual lab can take this job forward, given their limited bandwidth and limited expertise. Okay, this is really made possible by Glaucoma Research Foundation and also with the generous support from many of you sitting in this audience. So when we lump sum all this together, we're talking 30, 40 type of cells, and we can really have a zoomed in view to look into their not only their time course, but also their diversity. Okay, this is a linkage back to the 30-40 clusters I mentioned to you. Of course, this is only a subcluster of this, but this is like a hard work from two researchers across Stanford and UCSF over 2 years of time, where we identify who are the most susceptible populations and who are the most resilient populations, and what are their uniqueness in terms of morphology, genetics, but also importantly, what are the molecular protein out there, okay? So we're using this approach to look for unique proteins, to introduce the CRISPR tools, which you've probably heard a lot from the videos in terms of how RNA-based tools can come in and being carried and been delivered to eliminate endogenous proteins. And when we carry this, we can convert the resilient cells to a susceptible cells. But also reversely, when we introduce these proteins, in this particular case, it's a bone marrow protein that somehow expressed in this resilient population of cells called osteopontin, based on the name, is osteo origin. So we introduce this protein to the susceptible cells and show the resilience can be brought back again, okay? So all these are possible through the combination of the approaches and through the collaboration that we were working together, and really to think about how to have a healthy, effective adeno-associated viruses-based therapy for retinal ganglion cells, right? So all the success at the University of Pennsylvania through all the years of work focus on the outer retina, and can we have a similar relevant approach to do this at the ganglion cell level? And then I think we're at the very frontier to lead this effort. So I'm going to, because I'm using up my time, so I'm going to skip a lot of this, and I already told you most of the finding. And in this case, it's a resilient associated factor called osteopontin that is discovered, and through this collaboration, that will show they are very important to convert the susceptible population of ganglion cells and bring back to the normal conditions, and really show the possible regeneration. So and the next step, and then also obviously for those of you who are physicians and physician scientists in this audience, is really what is really the model, what is really the disease relevance. You know, we're generalizing this finding to other possible cell death related findings, and what is really going on, and what is really the spatial temporal resolution that we're dealing with. So, and also, as I mentioned many times, that this single cell genetic approach is really getting exploding, and it really bring us a lot of data. So a very talented MD PhD student, Nicole Tai, has been taking the single cell approach, really try to link the translation from mouse research to higher organisms, such as primates and human. And the reason this is getting very accessible is thanks to the genomics and genetics research, where an individual cell that is through the donor and through the careful characterization can give you a lot of information. Individual cell can carry the entire genome, the entire transcriptome, where the current resolution allow you to detect cell. So basically what we're doing is align our finding, including this osteopontin we discovered from mouse, and trying to align that to the ganglion type of cells that exist established in either primates, mostly primate at this moment, because the human data is very limited at this moment, and look for such evolution conserved populations. Are there Y cell and alpha cell equivalent in the monkeys that allow us to see this mirror image in vivo and to really bring this to the next level? So in parallel, and we are also very grateful for the Glaucoma Research Foundation support for us to recruit talented glaucoma researchers, residents, and physician scientists around us to work together. So taking this osteopontin example, so a very talented MD PhD, I mean, we should call him Dr. Kinde, and he was trained at Harvard and who moved to UCSF around the same time with me. And now he's a third-year resident, finishing his residency. Through the summer and the evenings and the weekends, and Dr. Kinde managed to work on this, on the pathological samples for human donors, and trying to find who are the osteopontin equivalent that exist in the human samples at different age, and what is really their physical relevance. And one of the dream experiments is to go back to the single cell genetics approach to really demonstrate you not only they share the same molecules, but also at functional level and molecular level, they're really evolutionary conserved. And this is a work in progress. Hopefully, by the time we can meet together in person in 2022, and some of the information can be more updated. And thanks to technology development at single cell level. So essentially, I think this current approach, as long as the sample is preserved relatively freshly, in the current technology can allow you to recover this information from human donors. And that is, I think it's a very, very powerful thing if you think about the technology development, excuse me, in the past 5 to 10 years, where a lot of the genetics and genomic information is really getting very, very accessible through this kind of very high resolution research. Okay, so last but not least, this is also Ben-Yan's work, where not only he can do the pathological research, but also he has been collecting aqueous humors from human patients. This is now at the log of 500. This data shows you 300 of those. Whereas through this surgery, he has been very carefully collecting aqueous humor samples, where we can use ELISA to detect osteopontin expression in the human samples. Okay, so at least at this moment, without any statistics, because this is a work in progress, we're just very excited to show you and hopefully to work with some of you, that we can show a correlation of up to 50% upregulation, even at the level of aqueous humor. This is not talking about the cell itself, but really the secretion of the molecules from the tissue to the fluid, right? So this is so. For those of you who are immunologists and for those of you who are thinking about this, we cannot justify this is purely from the ganglion cells. It's not just from the neurons, right? So there's many pieces of tissues that's connecting with this region. So that's a work in progress in terms of whether this is just a correlation or this is really a true finding that can lead to the next level. Okay. So and last but not least, there's a dream come true to do the gene therapy. And also, this is naturally linked back to all the mechanistic studies we're trying to tell you in the near future. Again, thanks again for the generous support. And I have already acknowledged most of the people in terms of their findings. And I would not waste your time to tell you what Anna and Derek have told you so far in terms of the stem cell-based reprogramming, and also in terms of the chemical screen of the inhibitors to regenerate. So the team has been working together, including this osteopontin work. And again, this is made possible through the Catalyst for a Cure Foundation, and that's reaching the 3 years old in a few months. And we're very grateful for your support and also for the guidance and the instructions from the board members and the scientific advisory board. Thank you for your attention.
Zen, thank you very much. Great presentation. And we really thank you and Meredith for what great work you're doing at the Glaucoma Research Foundation. I'm a layperson. I have glaucoma. I've had it since I was age 23, and I'm now age 70. So I've had great treatment. But I loved your discussion on population analysis, proteins, gene therapy, the optic nerve, and how you impact that, and also the single cell work you're doing. So I'm going to stop there and let the experts, the physicians, ask you questions.
Thank you so much.
Thank you, Steve.
Thank you, Steve.
And I know Dr. Emmanuel has joined us, and I've never been in a meeting he didn't have a question.
This is way over my head. Holy moly. I mean, this makes what we do look so easy. I actually texted Dr. Kornman on the side and was like, the complexity of the optic nerve is daunting, to say the least. I mean, we obviously don't learn about it in quite this detail in medical school or residency, but it's exciting to see this much detail, but it's also somewhat terrifying to see the complexity that we have to somehow solve to achieve our ultimate goal.
That's an excellent point, Dr. Emanor. So what's really believe it or not, throughout this presentation, we haven't even exited the eyes. The the optic nerve and the synapses, that's really a very active field of pursuing. So as you can imagine, all the effort, all the focus right now is really just this two-dimensional, oversimplified view of the ganglion cells superimposing on each other. But when you really think about how their nerves can be organized as cables of different colors, of different flavors, that really actually the eyes of different, goes to the different part of the brain. That map, that highway, however it is established and however we can rebuild it, that is really the frontier of the next phase. And I'm glad you pointed out. And really, when you think about this, the general finding is it's oversimplified. Is somehow the metabolically highly driven cells, and they generally have very big dendrites and very big axons and huge synapses. Those, in a correlated manner, has been the survivors subject to many of the glaucoma conditions. And what those cells generally carries signals such as adaptation and signals that's involved in the melanopsin, such as like basically the circadian controls. However, those very fine structures, such as those structures responsible for direction, structures responsible for this fine motion, those just generally just disappeared within a week or two, subject to these experimental models. So somewhere there's this fine, very infinite signatures that we really need to look into how to preserve it. Because I'm worrying a lot of the treatment, especially when it comes to growth, yes, we'll stimulate the growth, we'll keep the structure. But what happens is a lot of those very fine cells, even they are preserved, they'll be transformed. They don't know how to preserve their fine contact and fine synapses anymore. And I think our next frontier, our next structure, including a lot of work on this particular screen that has tiny bit of information, is really how to measure this. And we in the lab has been studying this is not even talking cortex, how the axon reach a subcortical area, in this case, tectum, right? The colliculus, the superior colliculus, one of the two prominent recipient area in the brain. Go back to your residency in medical school, and this is something subcortical. And for us, what does this mean? This means some looming, some fear, some like basic avoidance that has nothing to do with higher cognition. Somehow, this structure and these synapses are the most preserved, subject to silicone oil treatment, subject to glaucoma model. So that tells you evolution told us so much about it, and yet we're still learning. If we can have a way to, preserve all of them precisely and functionally, we will have a restoration of the functional vision. And that is really a joint force. So speaking of that, we are very, very grateful again for the Glaucoma Research Foundation, the board members and the scientific members of the advisory team that really guide us to the new frontiers. And this is really a dream come true. And it is not possible through the regular NIH funding mechanism to think these big questions closely related to human. Yeah.
Thank you, Zen. Especially to the physicians that are on, do you have questions related to what Zen just presented?
Meredith, I've got one. Will the team stay together in this next phase? You said it was funded and extended.
Great question. Yes. So the 4 primary investigators, Jen, Anna, Young, and Derek, will remain. And along the way, we may find different ways that we can, add some accelerator funding into what has already been planned for. But the 4 primary investigators will remain. So this is our 3rd catalyst for a cure. The first one lasted about 11 years. The second one about 8. So we plan on somewhere in the 9 to 10 year range. Typically in the first phase, it's getting to know each other, getting their labs set up and their teams set up. That's where we ramp up the funding. this team has blown away all expectations, and they've taken such a deep dive into the science already, not even at the end of year 3. So they're really ahead of the game. And as we move into phase 2, it's where we expect to see, the science to continue to unfold. There is a lot of talk around the fact that with some of the neuroprotection, that there might even be clinical trials, at the end of phase 2, possibly here. So I think we've got a great future ahead with this team, and it's really exciting. I think that the sky is the limit, and I think we're going to get really tangible results sooner than anybody expected.
Well, Meredith, we want to thank you, and we want to thank Zen for the great work of the Glaucoma Research Foundation. Are there any other questions for either of them? If not, Mike, I think we've covered our agenda. Do you have any closing comments?
No, I just want to thank Shin and Meredith for being here. We really appreciate your time, and you guys do a fantastic job of presenting your research and showing people what you're doing. And I think that is it really inspires us to want to support you guys and realize that there's people out there making progress on this, and that's good news. So thank you all both for being here.
Thank you. It's such an honor, and we thank you all for putting your trust and your resources in Jen and the team.